Hello students, this lecture is the continuation of the chapter Biological Classification. Following the Whittaker system of classification, the living world is divided into the 5 kingdoms. That was the accepted classification. So in this lecture, going to take up in details for the kingdom, means the first kingdom, kingdom Monera. Okay. So as we know, this is the first kingdom. Understood? The kingdom Monera includes the ancient, the primitive, smallest, and the simplest organisms. Understood, those primitive, smallest, simplest organisms should have a very simple organization. So means they are unicellular prokaryotes. Very important, these two words. Just repeating. Kingdom Monera includes the organisms which are unicellular, number 1. Number 2 is prokaryotes. As the name indicates, unicellular means only one-celled organisms are taken out from the kingdom Plantae or Animalia, because formerly the two scientists, Linnaeus and Aristotle, kept all bacteria in the kingdom Plantae. So, from the kingdom Plantae, the organisms which are unicellular and the one who don't have a well-defined nucleus taken out and kept in the kingdom Monera, or separate group is given to the organisms who are prokaryotes. Okay. So prokaryote stands for or means before, or a primitive, or a means pehle. Karyote means or karyon means is the nucleus. Means prokaryote stands for before a nucleus, means nucleus se pehle. So understood, nucleus is primitive or nucleus is not well defined, or it means prokaryotes means nuclear material of these organisms is not covered by or not within the nuclear membrane. Okay, that is very important. Just repeating again, kingdom Monera, the primitive group, include unicellular prokaryotes. Kingdom Monera is divided into mainly the two groups. Kingdom Monera is mainly divided into the two groups, that is number 1 eubacteria and the number 2 is archaebacteria. As again the name indicates, eu means true bacteria, archae means ancient bacteria. But in addition to the eubacteria and archaebacteria, one more small group, or not one more, it is the many other small groups are associated in this kingdom Monera. So only the one group is in your NCERT mentioned, just taking up that small group of the kingdom Monera is mycoplasma. So means we are going to take up the details for these groups, these 3 subgroups of the kingdom Monera. Just repeating, number 1 is eubacteria, number 2 is archaebacteria, and number 3 is mycoplasma, although mycoplasma is a small group. So eubacteria further includes the 2 groups, that is namely number 1 is bacteria and number 2 is cyanobacteria. Okay. A typical bacteria usually may or may not be a motile and bounded by a thick cell wall. And for mobility, they have a flagella. Blue-green algae usually are non-motile. They may be unicellular, may be colonial, or have a filamentous body form. That filamentous body usually surrounded by the gelatinous sheath. Can have a look in this video you have taken up. So next is taking up the details for the typical prokaryote cell. Means a typical prokaryote or a bacterial cell consists of cell envelope, cytoplasm, nucleoid, inclusion bodies, and for mobility have a flagella. In addition, have a pili or a fimbriae, but these two structures not help in mobility or in motility. So, one by one we will take up the details for these components of the bacterial cell, a typical prokaryote or a bacterial cell. So, students, in this chapter, this internal or a detailed cell structure of a bacterial cell is not mentioned. But mind you, in your school exams, when it is only even the first unit is coming in your exam, understood, they can ask you about the characteristics of the kingdom Monera and especially the characteristics of that a prokaryote cell. For that, you should know this topic, although this detailed prokaryote cell is in your third unit, the cell. But you should know even in this chapter the typical prokaryote cell characteristics, carrying the good a 5 mark with many of the objectives even in this chapter. Is that okay? Means this diagram is very important with the 5 stars. It is having the weightage in this chapter, in this unit 1. Okay, plus it's also having that same equally good weightage in the third unit of a cell also. In this topic, in this chapter, this question can be asked: what are the important diagnostic characteristics of the prokaryote cell or a typical bacterial cell? A bacterial cell or prokaryote cell के important features can be asked, just repeating a 5 mark question, or draw the diagram for a typical means in general होता है, for a prokaryote or a bacterial cell, having a very good important topic it is for this chapter also. So starting with the first component of it, the outermost covering of the typical bacterial cell is called as a cell envelope. This cell envelope is distinguished further into the 3 parts. The outermost layer is called glycocalyx or a mucilage sheath. Just repeating, glycocalyx or a mucilage sheath. So as the name indicate, mucilage मतलब ही क्या होता है? Slime, sticky. Mucilage sheath, if it is thick, it is called a capsule, and if the mucilage is loose, that sheath of the slime sticky layer is loose, it is called a slime layer. means glycocalyx may be called as a slime layer or a capsule. So slime layer or a capsule is followed by a cell wall. In the diagram just shown is a red color thick cell wall. The cell wall is not like that of plants, means not made up of a cellulose. Instead, the cell wall is made up of a chemical named as a peptidoglycan. Just repeating, can split up into the points, into the words. To memorize the word is peptidoglycan, CAN. It is a compound, chemical compound, just repeating, not of a cellulose. Instead, the cell wall is made up of peptidoglycan. The other name for the peptidoglycan is the murin. So that was the drawback for the two kingdom classification, if you can recollect. So the organisms whose cell wall is not made up of cellulose, why? That is, this group prokaryotes, means bacteria, is to be kept in the kingdom Plantae. Okay, this we have done already in the previous lecture of the biological classification, must go through. So is that okay? The cell envelope, the outermost coat is slime layer followed by a cell wall. So cell wall name indicate if it is a thick layer than the outermost layer, it gives a shape and provides the structural support to prevent the bacteria from burst or collapsing. Means it maintains the shape of the bacterial cell. The third means the innermost layer, from outside to the inside we are doing. The third innermost layer of the typical bacterial cell is called plasma membrane. Understood? You know, the cell membrane or a plasma membrane is thin and is semi-permeable in nature. Means it allows only certain molecules to enter into the cell, and it bears the respiratory enzymes. The cell membrane on inside bears the dots as shown in the diagram. It bears the respiratory enzyme. Means cell membrane not only perform the function of maintaining the shape, giving support to the cell, but in addition to that, it also performs the function of respiration, isn't it? Because it bears the respiratory enzymes. So still continuous with this is the cell membrane or a plasma membrane. Plasma membrane at a certain point shows the infold. Infold of the plasma membrane is called a mesosome. Repeating, a special membranous structure of the cell membrane is called mesosome. Means it is the ingrowth from the cell membrane or a plasma membrane. And this mesosome may be in the form of a tubules or maybe in the form of vesicles. It mainly help in the cell division, means cell wall formation or DNA replication, which is usually by binary fission in bacteria. And this point already I have taken up with you. It is the infold, so it help in increasing the surface area. Means it also help in respiration. Okay. Next is nucleoid. means it's not a term nucleus. In prokaryote cell, instead of the term nucleus, we use the word is nucleoid. Means DNA or nuclear material is not bounded by a is by the nuclear membrane. So it is called as a naked DNA. Nucleoid represented by single, circular, DS for double-stranded. DNA is double-stranded. This again we will take up in the third unit details for it. Just you have to go through this word just for this lecture. DNA usually is double-stranded in the higher forms, but even in a prokaryotes, in the bacteria also the DNA is double-stranded, means is DS representing here, DS DNA, double-stranded DNA. Naked term stands here for they don't have nuclear envelope. So that is for the next structure is the nucleoid. Definitely very important in the typical cell is to be drawn is the dots, dot means is representing the cytoplasm. For the dot, the term we use here is a granular cytoplasm. But a typical prokaryote cell do not show a streaming movement. Its cytoplasm do not show a streaming movements. And in a prokaryote cell, all the membrane-bound organelles are absent. Means a typical cell, a eukaryote cell, whether a plant or an animal cell, they have a organelles, might knows about Golgi apparatus, mitochondria, lysosomes, etc. But a prokaryote cell lack all the membrane-bound organelles, except for they have only the one organelle named as the ribosomes. In a prokaryotes, only the ribosomes are present, and that ribosomes is of the 70S type. That's why it is represented 70S type of the ribosome is representing here in a letter like an 8, like in a number is the 8. So 8 means this ribosome is actually having the two subunits, 50S and a 30S unit. 50S is a large unit and 30S is a small unit. So 30 and 50S units forms the 70S type of the ribosomes. Ribosomes, the main function for the ribosome is the protein synthesis, and for a protein synthesis 4 to 8 ribosomes usually. Just repeating, means many of the ribosomes they join with the help of a thread-like structure called a mRNA to form a chain called polyribosome or a polysomes. And these polysomes, they mainly are the site for a protein synthesis. Okay. Next, bacteria perform the function of photosynthesis. But students, as you know, for a photosynthesis, the organelle required is the chloroplast. But we are doing is the prokaryote cell. In prokaryote cell, except for ribosome, all organelles are absent. So in a prokaryote cell, instead of a chloroplast, we use the word is chromatophores. Because the bacteria or even the blue-green algae perform the function of photosynthesis, but we can't use the word is chloroplast here. Because in the chloroplast is present a pigment, chlorophyll. So they don't have a chloroplast. So coming to the next organelle or a structure, we can say, is chromatophores, which have a pigment, a photosynthetic pigment, and that pigment differ in the two groups of the eubacteria. So we have started with the eubacteria. Eubacteria includes the bacteria and blue-green algae. So here comes the point of a difference between the bacteria and the blue-green algae. So in the case of a bacteria, the photosynthetic pigment is of the primitive type named as a bacteriochlorophyll and bacterioverdin. This is the pigment of a bacteria, which help in performing the function of photosynthesis. But in the case of blue-green algae, the pigment is chlorophyll A, and that chlorophyll A is similar to that of green plants of the kingdom Plantae. Very important objective point here. What is the difference in the pigment of the bacteria or blue-green algae? And the next point, the next question can be asked is, why formerly, before the five kingdom classification, why the bacteria, they are kept in the, or kingdom Monera was not specified, why the bacteria was kept in the kingdom Plantae? Because in the blue-green algae, very important highlighting word is, in the blue-green algae BGA, the chlorophyll A is present, and that is similar to green plants, like that of photosynthetic plants, higher plants, they have a pigment. Okay. The next component of a typical prokaryote cell is the inclusion bodies. So inclusion bodies includes the structures which are non-living, not bounded by the membrane, and they lie free in the cytoplasm. All the 3 points is very important. Repeating: non-living structures, not bounded by the membrane, and they lie free in the cytoplasm. So inclusion bodies includes, number 1 is gas vacuoles. So gas vacuoles is again present only in the blue-green algae. Just repeating, gas vacuoles is present in the BGA, which consists of, actually, is the hexagonal gas vesicles, which help in maintaining the buoyancy of the cell. Because of that, if you know, they bring about, they here stands for the BGA, blue-green algae. Blue-green algae, which is bounded by the gelatinous sheath and are in the filamentous form, they usually brings about the blooms in the polluted water body called as the algal blooms, because of the presence of the gas vacuoles. Okay, your next point is, second is, under the inclusion bodies, inorganic inclusions. Inorganic, name indicate, inorganic elements is the sulfur or a phosphorus. So they may have in the form of a granules called the volatile phosphate or a sulfur granules. Next is the food reserve material. Here again the point of a difference comes between the bacteria and a blue-green algae. Of the topic is the eubacteria. So blue-green algae is like in higher plants. They have a food reserve material in the form of a starch. Also can be called as a cyanophycean starch, because the other name for the BGA is cyanobacteria. So again the bacteria, in the cyanobacteria, the food reserve material is like that of a higher plants. It is a starch, and even they have a pigment like in a higher plants, chlorophyll A. But in the case of bacteria, the food reserve material is in the form of a glycogen, or as you know, glycogen is the food reserve material of a animal cell. But typical, general, most of the bacteria, they have the food reserve material in the form of a glycogen granules. Next is about the typical bacterial cell may be motile or a non-motile. If they are motile, they have a filamentous extension. That filamentous extension of the bacteria from the cell wall coming out, it is, we can say, called as the flagella, which is made up of protein flagellin. And a typical flagella consists of three parts. Number one is the basal body, which is embedded in the cytoplasm. Next, the part which is covered or which is within the cell envelope called as a hook, and the exposed outer part is called as the filament. Is it okay? This help in mobility or motility. Usually in the motile forms of the bacteria is present a flagella. In addition to the certain bacteria, they may have a pili or a fimbriae. These two structures, pili and the fimbriae, they not help in movements. Pili are elongated hollow structures. They help in reproduction and made up of protein pillin. While fimbriae, they are bristle-like and they help in attachment. Okay.